It is lesson 6 this time, and we're going to discuss about carnivorous plants. Plants have got the fascinating ability to eat other creatures. Some plants aren't eaten by bugs. They eat the bugs themselves. Nearly 700 species of these carnivorous plants live throughout the world. A hungry fly darts through the pines, drawn by what seems like the scent of a nectar from a flower-like patch of scarlet on the ground. The fly lands on the fleshy pad of a ruby leaf, and it takes a sip of the sweet liquid oozing from the leaf, brushing a leg against one tiny hair on the surface. Then another, suddenly the fly's world then falls around it. The two sides of the leaf are closing against each other. Spines along its edge interlock like the teeth of a jaw trap. As the fly struggles to escape, the trap squeezes shut more. Now, instead of offering sweet nectar, the leaf unleashes enzymes that eat away at the fly's innards, gradually turning it into goo. Carnivorous plants, sometimes called insectivore plants, are any plant specially adapted for capturing and digesting insects and other animals by means of ingenious pitfalls and traps. Carnivory in plants has evolved independently about 6 times across several families and orders. There are more than 600 known species of carnivorous plants, constitute a very diverse group, in some cases having little more in common than their carnivorous habitat. Carnivorous plants are plants that derive some or most of their nutrients, but not energy, which they derive from photosynthesis, from trapping and consuming animals or protozoans, typically insects or arthropods. Carnivorous plants have adapted to grow in places where the soil is thin or poor in nutrients, especially nitrogen, such as acidic bogs. True carnivory is thought to have evolved independently 9 times to 5 different orders of flowering plants, and it is represented by more than a dozen genera that attract, trap and kill prey, absorbing the resulting available nutrients. But there's always this question: how did they evolve? The evolution of carnivorous plants is obscured by the paucity of their fossil record. Because they are actually soft, very few fossils have been found, and then usually only as seed or pollen. Carnivorous plants are generally herbs, and their traps are produced by primary growth. They generally do not form readily fossilizable structures such as thick bark or wood. Most carnivorous plants, however, live in habitats with high light, waterlogged soils and extremely low soil nitrogen and phosphorus, producing the ecological impetus to drive nitrogen from an alternate source. It has been suggested that all trap types are modifications of a similar basic structure. The hairy leaves can catch and retain drops of rainwater, especially if shield-shaped or peltate, thus promoting bacterial growth. Insects land on the leaf, becoming mired by the surface tension of the water, and suffocate. Bacteria just start decay, releasing from the corpse nitrates that the plant can absorb through its leaves. This foliar feeding can be observed in most non-carnivorous plants as well. Plants that are better at retaining insects or water, therefore had a selective advantage. Rainwater can be retained by cupping the leaves and pitfall traps, that for example in pitcher plants, and may have evolved simply by selection pressure for the production of more deeply cupped leaves, followed by zipping up of the margins. And most of, for example, plants such as the Venus flytrap have hairs. Only the insects can retain by making the leaves stickier by the production of mucilage, leading to flypaper traps such as Sundews. So the conspicuous trapping mechanism, which is always a modified leaf, draws special attention to these plants. A variety of trapping mechanisms exist and are designated as active or passive based on whether they move to capture prey or they are still. These traps may be active or passive depending on whether movement aids the capture of prey. For example, Triphyphilum is a passive flypaper that secretes mucilage but whose leaves do not grow or move in response to prey capture. Meanwhile, sundews are active flypaper traps whose leaves undergo rapid acid growth, which is an expansion of individual cells as exposed to cell division. The rapid acid growth allows the sundew's tentacles to bend, aiding in retention and digestion of prey, which you actually saw in the video itself. There are actually 5 basic trapping mechanisms that are found in carnivorous plants. Flypaper traps use a sticky mucilage. Pitfall traps, such as pitcher plants, trap prey in a rolled leaf that contains a pool of digestive enzymes or bacteria. Snap traps utilize rapid leaf movements. Bladder traps suck in prey with a bladder that generates an internal vacuum. And lobster pot traps, which are very rare, also known as eel traps, force prey to move towards a digestive organ with inward pointing hairs. So we'll be going in each in detail. Pitfall traps, flypaper traps, snap traps, and bladder traps. With the plants, this is a bladderwort plant, pitcher plants, sundews, and Venus flytrap. Or tropical pitcher plants of the genus Nepenthes. In the 100 of the so species of this genus, the pitcher is born at the end of the tendril, which grows as an extension to the midrib of the leaf. Most species catch insects, although the larger ones, such as this one, which you can see a mouse next to it, the Nepenthes raja, also occasionally take small mammals and reptiles. Nepenthes bicalcerata possess 2 sharp thorns that project from the base of the operculum over the intestines of the pitcher. These likely serve to lure prey into a precarious position over the pitcher mouth, where they may lose their footing and fall into the fluid within. Flypaper traps, such as the one which we saw in the sundews. Flypaper traps can be active or passive and rely on sticky mucilage either directly on the leaf surface, such as the butterworts, or on gland-tipped hairs, sundews, to capture prey. The flypaper traps utilizes sticky mucilage or glue. The leaf of flypaper traps is studded with mucilage secreting glands, which may be short like those of a butterworts or long and mobile like those of many sundews. Flypaper traps have evolved independently at least 5 times. The sundew genus Drosera consists of over 100 species of active flypaper traps whose mucilage glands are born at the end of long tentacles. However, the one such as the snap traps which we saw in the video that prognates to cells in the midrib. This whole process takes less than a second. Actually, once the electric impulse is sent to the plant closes. You know, in the Venus flytraps, for example, closure is response to raindrops and blow in debris in prevented by leaves having a simple memory, you can say. For the lobes to shut, 2 stimuli are required to be triggered between 0.5 to 30 seconds apart. However, bladder traps are only found in bladderwort plants and are actively sucked in small organisms using a partial vacuum. Bladder traps are exclusive to the bladderworts. The bladder will pump ions out of their interiors. Water flows by osmosis, of course, generating a partial vacuum inside the bladder. The bladder has a small opening sealed by a hinged door. In aquatic species, the door has a pair of long trigger hairs. Aquatic invertebrates, such as small crustaceans, touch these hairs and deform the door by lever action, releasing the vacuum, and on that time the invertebrate is sucked into the bladder. In all these type of traps, by using enzymes or bacteria, carnivorous plants digest their prey through a process of chemical breakdown analogies to digestion in animals. The end products, particularly nitrogenous compounds and salts, are absorbed by the plant to enable their survival under otherwise marginal or hostile environment conditions. Most carnivorous species are green plants to enable them their survival, so they actually do photosynthesis and they manufacture their own food. But in Venus flytraps, such as this one, which is Dionidae muscipula, its traps are much more different, because they have actually got more foraging, flying or clawing insects such as flies are attracted to the cavity formed by the cup leaf, often by visual lures such as anthracynid pigments and nectar brines. The rim of the pitcher pristome is slippery when moistened by condensation or nectar, causing insects to fall into the trap. Many carnivorous plants have a special glands that secrete enzymes powerful enough to penetrate the hard exoskeleton of insects, so they can absorb as much as nutrients from the inside of their prey. But the purple pitcher plant, which lives in bogs and infertile sandy soils, enlists other organisms to digest its food. It is home to an intricate food web of mosquito larvae, as I said earlier, midges, protozoans and bacteria, many of which can survive nowhere else but only in the unique habitat of the pitcher plant. So there are actually very unique species that you cannot find anywhere else but inside the pitcher plant itself. The animals shed the prey that fall into the pitcher, and the smaller organisms feed on the debris. Finally, the pitcher plant absorbs the nutrients released by the feeding frenzy. It's actually amazing to see how different plants, different species can actually coexist with each other like this and have this fascinating relationship. Now, the sundews. There are approximately 152 carnivorous plants and catch its prey in less than a millisecond. Thank you for joining this lesson. Hope you enjoyed, and make sure you click on that subscribe button, like and share, so we can reach a higher audience and of course be able to reach more people and spread awareness and help them learn about the natural world. Be curious, love the natural world and protect it. Thank you for watching this video. If you like it, please give us a like, comment and also share. And don't forget to subscribe and hit on that bell icon so you can follow us through all our adventures and research.